The optic nerve, a cable-like grouping of nerve fibers, connects and transmits visual information from the eye to the brain. The optic nerve is mainly composed of retinal ganglion cell axons. In the human eye, the optic nerve receives light signals from about 125 million photoreceptor cells via two intermediate neuron types, bipolar and amacrine cells. In the brain, the optic nerve transmits vision signals to the lateral geniculate nucleus, where visual information is relayed to the visual cortex of the brain that converts the image impulses into objects that we see. In the retinal tissues of the eye, more than 23 types of RGCs vary significantly in terms of their morphology, connections, and responses to visual stimulation. Those visual transmitting RGCs are the neuronal cells. They all share the defining properties of possessing a cell body at the inner surface of the retina, having a long axon that extends into the brain via the optic chiasm and the optic tract, synapsing with the LGN. The RGCs form multiple functional pathways within the optic nerve to mediate the visual signal. Human beings can see three primary colors: red, green, and blue. This is due to our having three different kinds of color-sensitive cone cells: red cones, green cones, and blue cones. The RGCs connecting to the red and green cones are midget RGCs. They are mainly located at the center of the retina, known as fovea. A single midget RGC communicates with as few as 5 photoreceptors. They transmit red-green color signals to the parvocellular layer in the LGN. The midget parvocellular pathway responds to color changes, but has little or no response to contrast change. This pathway has center surround receptive fields, and slow conduction velocities. Because of this pathway, we can see objects precisely in detail and in full color. A fully functional optic nerve is essential for vision. Obviously, any damage of the optic nerve will sever the precise transmission of visual information between the retina and brain, directly leading to vision distortion and/or vision loss. Damage to the optic nerve can result from direct/indirect physical damage, acute/subacute physiological lesion, chronic neuronal degeneration. Moreover, the optic nerve is also a very important vivo model for studying central nervous protection and regeneration. At the cell biology level, the RGC axons are covered with myelin produced by oligodendrocytes after exiting the eye on their way to the LGN and thus part of the central nervous system. Scientists have recently acquired more and more evidence that certain types of damage to the optic nerve may be reversible in the future. Therefore, the optic nerve provides a potential window to explore more complicated neuronal degenerative diseases, such as Alzheimer's disease and Huntington disease.